Okay, welcome everybody, and thank you for taking time to listen to our presentations today. I'm going to start out with COVID-19, the pandemic, evolution, and epidemiology. Learning objectives are going to be describe the anatomical characteristics of a virus, review viral mutation, describe the evolution of the COVID virus, and then look at some of the global consequences of COVID-19. So to start out with, I want to thank all the healthcare workers, and I know some personally because I've worked with these individuals. What a great job you guys have done at the bedside. You've risked your lives. You've done unbelievable, great work at the bedside, and I want to just again praise you and laud you and many other adjectives for being true heroes at the bedside. So when we look at a virus, we look at general characteristics of the virus, okay? To start out with, viruses are obligatory intercellular parasites. And this is very important to understand, that viruses cannot survive on their own. They need some kind of host. They either contain a DNA or RNA, which is kind of the brain of the virus. Some contain a protein coat. Others are enclosed by an envelope. Some viruses have spikes that allow them to attach to the host tissue and be able to replicate rather easily. Most viruses infect only one specific type of cells in a host. As we know with COVID-19, that virus enters through the nasal passages and through the oral mucosa, but it can infect multiple other cells once it enters the host. And host range is determined by specific host, attachment sites, and cellular factors. Now, when we look at this slide here, you can see the different types of viruses. And a lot of these viruses have the characteristics that I alluded to. If you look at the HIV virus, you can see these are the protein spikes. The bacteriophage has actually pointed spikes. So all these viruses here have have the characteristics that I described in the previous cell. And, a lot of them, use very, very sophisticated ways of attaching to host cells and being able to replicate. So if you look at an anatomy of a virus, so if we take a virus and we slice it apart, this is kind of what a virus looks like. And again, when you look at the nucleoprotein, the RNA, this is kind of the powerhouse of the cell, in a sense, like the mitochondria for our tissues, where, this is the cell, this is how they can replicate, and this is the actual information or genetic code that allows the cell to be able to replicate. Then we have this lipid envelope that helps protect the virus. So this helps the virus be protected by the host immunity response, or by maybe medications or other type of interventions that would destruct it. The capsule, this green kind of envelope here, or green kind of matter of the cell, it helps protect the nuclei from digestion of enzymes, and it uses the virion to penetrate. So this secretes a fluid, if in the virion, that can go into the spiked cell, that can help cause toxins to go from the virus to the host cell. All right. So it can inject some of the nucleic acid into the cell's cytoplasm. So you have this normase, which is kind of a spike that is responsible for how it attaches and spreads to the host. And then you have the hemagglutinin, which releases the virus antibodies in the host. So this is kind of a typical anatomy of a virus. And again, this is how it's going to replicate. This is how it's going to attach. This is how it's going to cause the virus to become toxic to the host. And these over here are going to be how they protect the virus from getting destroyed. And when we talk about treatment, this is how we have to think about how can we treat this virus? How can we destroy it? So when we do treatment, how can we, what can we do to make this virus unstable or weakened? And again, this is just the COVID virus, which again is very similar to a true anatomy of the virus. It has an RNA, it has spike proteins, and it has a lipid membrane. This is a pretty interesting, shows how quickly these viruses can replicate. This is human tissue in a lab, and the COVID virus has been injected into this tissue, and within about 1 to 2 hours, you can see how dramatically the virus has replicated from just 1 or 2 actual injections of the virus. So it shows you that this doesn't take long to replicate, especially the COVID-19 virus. And this was before the Delta mutation. Now, one of the good things about viruses, and you know, we haven't found many bright stars or shining lions during this pandemic, but one thing is that viruses are relatively easy to kill. If you look at a hierarchy or pyramid of destruction or the ability to destroy these parasites or organisms, you can see that viruses are very low on the bottom there. And then you have vegetative bacteria, fungi, mucobacteria, and the hardest to kill is either protozoa or bacteria spores. So luckily, at the very bottom of this pyramid is the actual viruses, and they're the more susceptible to destroying. So if this pandemic was caused by bacterial spores, the majority of us would probably be dead by now, because it'd be very, very difficult to control. So when you think about people that have C. diff and things like that, of how hard it is to protect yourself, you just can't use hand sanitizers. You have to use soap and water for, you know, a period of time. So when we look at just what kills viruses, so, you know, we look at soap is very interesting, because we don't really think about when we wash our hands what's happened to the virus. So again, you know, we have the spike protein that helps enter, you know, the cells, lipid membrane that helps protect, and here's the genetic material. So when you look at soap molecules, okay, they bond with water and they kind of go in there, and the water shuns the tails of the virus, and then causes the virus to kind of kind of separate and kind of fall apart, disintegrate. And at the same time, it traps the spikes that would normally be transmitted. The bubbles go around it, and then obviously they wash away in water. So soap is very, very effective against these viruses, because it helps, you know, from the friction of scrubbing, destroy the virus, and but at the same time, it traps the kind of a cell, you know, the virus attachment, and then it goes down the drain. Hand sanitizers aren't quite as effective, but the idea is that they, you know, they need to be at least 60%. And what they do is they help, again, penetrate through the envelope and kind of cause the alcohol to destroy the cell wall and membranes of the bacteria cells and viruses. They're less effective against non-envelope viruses, but again, they have to be a minimum of 60%. And again, if your hands are very soiled and they have a high viral content, these will not work as effectively as soap and water. But they do cause cell disruption, cell membrane disruption, which then allows vulnerability to the viral genome. And again, these are, again, different types of sanitizers, cleaners, disinfectants that can be used in different ways. So you look at, again, a viricide kills viruses. So that's kind of what you want to focus on. Again, some of these things will just aid in soil removal, but you really, if you get viruses on surface, you really would kind of like to either use a viricide or a sterile lantern in order to kill the viruses, not just soap and, not just in a sense of soap. If they get on, really entrenched into material or on, ventilators and things like that. And again, these are very common. They're all over the place now. Back in March and April of 20, you couldn't find them if you would have paid millions of dollars for them. Now, the question always becomes how the viruses spread. They can either be droplets by cough and sneezing, that can spread droplets of saliva and mucus. A lot of these are large. If they're like 5 microns or more, they will end up not going into the lung. They will end up going down and dispersing before it gets to the airway. Airbornes are tiny particles produced by talking and suspended in air for longer time periods and travel further. And they're smaller, so the chance of inhaling them is greater at that point. So it's not that obviously you can't spread disease by droplet. That's the typical influenza virus, but COVID being airborne makes it much more susceptible for transmission because those particles stay suspended in air and they can stay up there to be inhaled for longer periods of time, and they can also transmit a longer distance. And I think this is a very nice picture of what the viral transmission can be on somebody who coughs or sneezes. Okay, I mean, you can see that we normally don't see this. We only see if someone sneezes out or coughs out a rather large specimen, but you can see that with this individual there is a whole plethora of transmission generated from this one cough. And it gives you an idea too. So this is using a black light on somebody coughing and how far the transmission of that cough is. It's, this is 7 to 8 meters. So that adds up to a fair distance, almost 6 feet or more. And you can see that there's within the half of that, so within the 4 or 5 meter range, okay, there's a really large plume of transmission. So I mean, this has been the argument to use masks because you can protect, 1, yourself, and 2, people can't disperse this all over the air. And again, when you look at, so no mask, this is a transmission, and this is with a tightly worn non-woven mask. And you can see there is some transmission that does escape, but not anything comparable to the transmission that occurs in the unmasked individual. And this is an example of viral transmission. So again, using a black light, and this was vaccinated students in China who they had COVID shot into the air in this experiment, or whatever you would call this clinical trial. And with a black light, it showed where the transmission and where the viruses actually were sitting and dispersed just from talking with individuals that were unprotected. And you can see that it's in food, it's on this person's hands, it's on this person's face, on their glasses there and over there. So you can readily visualize that the transmission of this virus is quite dramatic, both from the dispersion in air and from some contact on when someone has just recently coughed, sneezed, or talked. So again, where do you get COVID? Crowded places with many people, closed contact settings, okay, and confined spaces with poor ventilation. So again, you argue the point that you stay away from people and you stay outside, you probably have less chance of getting COVID than if you're inside with lots of people, especially if they're unvaccinated and unmasked. So the treatment for COVID or for all these is really prevention. For COVID, we have no magic bullet. I know we've tried all kind of arrays of interventions, anywhere from using dexamethasone, ritonavir, all these other type of concoctions, and there really is no outcome to say outcomes are any better with one type of intervention than the other. The only thing we have is really vaccination. Now, some viruses we do have antiviral drugs like Tamiflu and Lenzavir, which is a spray inhaler to be used with asthmatics. But typically, when you get a viral infection or a viral case, it's really more supportive rest fluids and anti-inflammatory drugs, and you try to hope that either your immune system kicks in and helps you get better, because again, for COVID-19, we have no magic bullet. We do have some antiviral drugs out there for other viruses that I'll cover in a second. So when you look at the influenza virus vaccine, so you look at influenza, there is both the live vaccine version and the actual non-live egg grown at this point. And there are different types of options to be able to be administered to different groups of individuals based on age and comorbidities. As you can see, like someone like me who's just about 67, I got the flu vaccine this year. I got the trivalent, which is an actual adjunct is added to the vaccine to make it more effective in people. All right, and you can see that some of the strains are live, and the nasal vaccine can be used for children or even young adults. So there's different ways to administer vaccines historically. I know with the polio vaccine, actually a sugar cube has been used. Now we do have antiviral treatment for other viruses, and we'll talk about it. We have the antimdine, which blocks the blocks influenza virus by encoding entry into the respiratory cells. So this basically helps protect the respiratory tissue and makes it more difficult for the virus, the influenza virus, to attach to the respiratory mucosa. Only affects influenza type A. And the other two blocks the activity of viral neuraminidase, which is really the spike protein that attaches to the host tissue. And most are familiar with this one. This is the scientific name for the generic and for the brand of Tamiflu. Okay, and what's really important is that if you're going to use these antiviral drugs, and it's really the same thing with COVID. I mean, they are working on antiviral COVID medications, but again, it's not going to be beneficial if you utilize it like 5 days into it. It's most effective started within 48 hours of onset and of symptoms. It can be reduced the duration by 1 or 2 days. Okay, so we do know if you use these early on, you can have a reduction in the duration of it. But again, it has to be used early on. So a minute you have symptoms of the flu, it needs to be given. And I think that's one of the big problems we've had with COVID is a lot of times we give the anti-inflammatory agents like ritonavir and dexamethasone after the individual with COVID is sick and they're hospitalized. They really need to get that right when they're tested positive with COVID. Some of these things should be administered in order to improve mortality and morbidity. Now, when we look at Tamiflu, I think this is rather interesting the way this works. So do you see here, Tamiflu puts down this green spike. And what the virus is doing is putting down these blue tentacles. And in order to replicate the virus to attach to the tissue. And what happens here is Camel flu, the green spike, fools the virus, and the virus attaches to this versus to really what it's actually transmitted to the host tissue. And when it hits this antiviral medication, it causes the viral to disintegrate and disrupt, and thus it cannot replicate and spread throughout the human tissue. This is lung tissue there. So it's really nifty the way scientists and researchers figured that out, that it actually fools and confuses the influenza virus. Now, don't do crazy things. During this pandemic, it was said that if you drink ethyl or bleach, or you soak yourself in a bleach bath, and this will kill the viruses or not. We do not have any method that if you take internal antiviral solutions that will have a improved outcome with COVID-19. They're methyl, ethyl, and bleach are poisonous, and drinking them can lead to large disability and deaths. And there was a whole group of people who got this ideas, and they actually did this, and they ended up either unfortunately dying or being very, very sick. So again, bleachers work on the outside surface and work on your hands, but they don't work on your internal organs, especially your lungs. So when you look at the COVID vaccine, and Terry's going to talk about this, understand how these vaccines do work in the sense of the cell. So when you look at RNA vaccines, what they're trying to do is they're trying to tell cells to produce the spike protein. So what it's doing is it's going to fool the cells. And as the virus comes in, the antibodies and T cells are going to recognize this virus, and they're going to attach right on to the coronavirus and not allow it to spread. Okay. So again, the idea with the RNA type of vaccines, and Terry's going to go in more detail with this, so I don't want to belabor it. Okay, is that you again try to mimic the virus, and your cells recognize the virus, and immediately your antibodies and T cells come to the rescue and attack this virus. And it appears that the vaccines do very, very good, the RNA vaccines, in the sense that the individuals that have got vaccinated who have positive testing for COVID have very minimal hospitalization and basically no death at all compared to unvaccinated individuals. And then there's the viral vector vaccines, which actually use some of the virus and actually goes in, and what it tries to do is replicate the virus in your body to get an immune response. So when it sees a high dose of the virus, it will respond appropriately to your body. So the idea is you're going to get a little bit of a immune response, but the second time, which will be the virus scan into your body, you will actually have a much higher immune response, which will prevent the virus from going on. And these are the ones that are out there. And again, I don't want to, take Terry's thunder. There may be more out there, but again, most of the doses have been 2, and just tells you the type of they are. So there's a combination of RNA, and there's a combination of viral vectors, and then you have basically how many doses you need and the storage. And the storage is a real problem with the RNAs. You can see it takes a tremendous amount of temperature in order, especially the Pfizer. It's minus 80 to minus 60. So again, it's trying to store these vaccines can be a tremendous task in itself. And, again, when we talk about global herd immunity, I don't think we're going to be able to reach that because this is understanding that only like 1% of people in low income countries have received at least one dose. So the problem is, there's, you can do 70% of the US have been vaccinated. All right. But the world in general, less than 10%. So the idea that you're going to get global herd immunity because of vaccination distribution, that is not possible. So just keep that in mind. Now, you look at, I just throw this out here, like a country of Somalia. They have a total population of 15 million people. And as of end of January, they only had 200,000 people vaccinated and only 1.3 of it. So, and again, we talk about these things called vaccine deserts, where, and we see this in our own country, where you have not a pandemic anymore in the United States of COVID-19, but more like an endemic, which is saying that only certain areas are having surges while other areas are having minimal, a surge. So you see this through the world where, you may be able to get control of the pandemic in your more industrial nations, but to try to get control of the pandemic globally is almost impossible, if ever achievable. And that's something to keep in mind, because as long as the virus still survives, it always has the potential to keep on mutating into a more more lethal, more transmissible type of virus. So again, you look at this and you can see that the vaccine has not been equally distributed for many reasons. And in Africa, we have no data at all. So, I just bring this up when it comes, they're hoping that by 2023, they can finally get the vaccination into Africa, but that's 2 years from now. And like, who knows? So the idea about, being able to get herd immunity is kind of questionable. Now, some have thought about mixing the vaccines, and some data came out saying that the vaccines is not a good idea to mix, that there seems to be more side effects when you mix a RNA vaccine with a vector vaccine in the sense of side effects. So I'm not sure if that's something that's going to be advocated. I mean, I did hear recently about, individuals that got Johnson & Johnson, which is a vector vaccine, to get a RNA vaccine as a booster. But I'm not sure where we're going to go with that, because it appeared to be no more effective and it appeared to have more side effects. Now, again, ritonavir was approved to treat COVID-19, and it was really developed in the sense of trying to treat Ebola and SARS and MERS, and it became a rather quick standard treatment for COVID in many hospitals. And a limited trial showed that some patients recovered more quickly with it, but it didn't show to prevent any disease. But now when you look at it, there's no evidence that COVID-19 outcomes were improved by ritonavir. I mean, it's still given, but the problem is that it's given too late. The idea is it stops the virus from replicating. It could be more effective earlier in someone's disease before the virus is spread through the body. But ritonavir, because it was approved as an emergency drug for COVID, is not given in the outpatient arena. And thus, I think it's used inappropriately, unfortunately. There is a work on a COVID pill that is looking at the same way that the Tamiflu works. Okay, Pfizer's looking at a pill, and so is Merck, to try to target the enzyme key to viral replication. So there may be in the future, and this was talked about, maybe the end of this year or early 2022, that there could be a treatment for COVID, just like we have Tamiflu for seasonal flu. Now, the one problem is that when you look at flu virus mutations, a flu virus mutation can mutate 413 times in a given season. Human DNA has only changed 1% in 40 million years. And a flu RNA can change 1% in 4 days. So avian flu, when it gets to humans, can be highly pathological and can be responsible for 50% deaths. So one of the things that we really need to, or one of the concepts, and really we need to put our hands around when we deal with flus, they're going to mutate. They're going to mutate. Okay, so keep that in mind, that you can't stop viruses from mutating other than if you totally prevent hosts from getting viruses. So the only way you can prevent the current COVID-19 from mutating is not to have people infected with it, because once someone gets infected with it, the virus survives and thus lives for another day to change. They're changing all the time. This is why one person can get the flu repeated times. You can get the flu every year. You can get a cold every year. All right. You don't get measles every year. Okay. Because you have a vaccine that can, and measles, they haven't, that hasn't changed their DNA, but viruses change all the time. And again, there's great fears. All right. This is an example of this avian flu, where a bird is actually the carrier of the influenza strains, and then it affects a pig, which is considered to be the intermediate host, and then that pig comes in contact with the human and thus transmits this. The bird doesn't infect the human, it actually infects the pig, which then infects the human. All right, and this is a woman that's obviously freaked out because her child is playing with a dead bird who could be carrying avian flu. Now, this is a slide that I had back in presentations I did almost a decade ago, okay, after swine flu, H1N1. And the point was, there was always a future of flu fears. I said in many presentations that people worry about global warming. I think it's a big concern. People worry about a nuclear war. There's a lot of crazy leaders out there. And 3, there's worrying about the future in general. My concern always was that the fear of viruses, that viruses could take over and could decimate humanity. And what you see is with this picture here is that things can stay rather stable when it comes with flu, and then all of a sudden it can spike up dramatically. So it's very, very important to realize that all you do need is one mutation of a virus, and you can spike the amount of deaths considerable. You look at cancer, which is very, very stable, and all of a sudden, it doesn't change that much. But you look at the change in, you get a swine flu, and boom, it goes up dramatically. Now, I hope we never had this kind of mutation, because then it's time to probably kick in and give up. So why do viruses mutate? All viruses, including the one that causes COVID-19, mutates. We know this now. These kind of viruses are very, very complex and very smart. In order to live, they know they have to be one step ahead of the immunity of the host, and they need to be able to replicate themselves in order to thrive in different type of environments, different type of weather, different type of spacing, and different type of human beings. Okay, and immunity response. Okay, and not all mutations are beneficial for viruses. Sometimes viruses mutate, which make them more, more easier to destroy, or they can kind of become too virulent and they destroy their host too quickly and they can't spread. Okay, but some can make more infectious and threaten to the host humans. And unfortunately, each time COVID has mutated, it has become more lethal, and it has become more transmittable. So it's somewhat concerning that this virus could be extremely ingenious on how it mutates. Now, what I'm saying here is, so this is your COVID spike protein, okay, and you can see the design of that. So as it starts mutating, it can change its opening in the way spike proteins go. And here you have 3 openings compared to the one here. All right. But that also can leave the virus more vulnerable in order for the immunity to get to the virus nucleus and the RNA to prevent it from replicating and become more easier to destroy. So again, when these viruses mutate, they do take the risk that they could be more vulnerable to antibodies of the host, and also things like vaccines. But usually when they mutate in this sense, when they go 1 spike, 2 spike, 3 spike, the likelihood of transmission increases dramatically. And this is what we've seen with COVID-19. There are 3 types of variants circulating at this point. Okay, so variant B1.1.7 was the first detected in September of 2020 in the United Kingdom. Then we had B1351 in South Africa, called the South African, and then there was one also in Brazil at this point. So again, this was before we had the Delta. So we had evidence of mutation within about, so you would argue the point that Wuhan probably started with this virus in October of 2019. So within 1 year, there was evidence that this virus had mutated at least 3-4 times. And again, we had, they all again seemed to increase their transmission, and they were stickier due to the changes in their spike protein, which made the transmission even greater. So again, each one that each time it increased its, each time it mutated, it increased its transmission by changing its structure. May it make its spike protein more stickier, so it adhered easier, or it had greater amount of spike proteins, which then give a greater amount of adherence to the host tissue. So again, this is kind of what I said. Mutation 1 could mean that it increases transmission, but it causes them to be somewhat, more susceptible to intervention. And 2 could be very worrisome mutation. And then quite 3 is it changes itself and could actually totally be recalcitrant to any type of vaccines that we have currently. And this again, I have slides from the whole pandemic. So this was as of March 2021, when we looked at the different, so the UK variants you could see was, sparse throughout the United States. Certain areas, I remember Michigan was hit very hard by the South African variants, but the rest of the country didn't see a huge surge. Florida, they saw a surge because a lot of people vacation down there during the spring. But the South Africa and Brazil made very minimal impact in the United States. Now, the current one is the Delta, which has a triple mutant COVID variant from India, and it's a global health risk. And this was back in mid-spring, right? And they call this the B1617. And this variant, they were very, very concerned because there were in India 3,879 deaths per day in India, and there was almost 400,000 new cases in 7 days. All right. And this variant started to spread to other countries, including the United States, but obviously it didn't really become robust in the United States until last month. And you could see, the effect where you had tremendous amount of individuals in India who were living close quarters, who were waiting for some kind of medical care. This is the emergency room at one of the Indian hospitals, and this is the line outside of people waiting to be treated. None of them look too bad at this point, but all of them have symptoms of COVID, and they're trying to be tested for it. There was a very shortage of oxygen and other compressed gases, and individuals lined up and kind of, for lack of a better word, claimed their own cylinder of oxygen so they would have it for their family because they could not count on institutions having it. This is a very grim scene. In India, when someone dies, they need to be burned within 24 hours. And this is an area where they have all these individuals stacked with wood, and they're going to burn them. And at one point, they were actually running out of wood because they were burning so many bodies. They were burning more bodies than they could actually accumulate wood and cut down trees and stuff like that in order to use for these burials. So it's very, very grim in India. And then we got these things called these fungus infections, which I don't think we've seen in the United States, but it was known as black fungus, saying that individuals were actually not only getting respiratory symptomology from Delta, but they were actually getting blood clots in the optic vessels that were causing individuals to go blind and losing function of their eye. And this is what it looks like. Again, this is a clot in the actual vessels leading to the optic nerve. Now, this was in the middle, so this was the end of June, so almost the start of summer. And you can see that, the Delta variants, it was much more pronounced compared to when we looked at South Africa and we looked at the, England variants. As you could say, they were kind of sparse, but within a short period of time, especially the Midwest, had an increase in Delta. And unfortunately, this has increased dramatically in non-vaccinated areas. So they actually, this was last week, that new cases are up by 140%, hospitalizations are up 34%, and deaths are increasing by 33%. And doctors are saying, if you're going to go to Florida, you're at very high risk, especially if you're unvaccinated, because the transmission of this and the areas in the darker, the red, are the higher areas. So my poor son is a policeman in New Orleans down here, and they're totally surged out. He was telling me that when he calls for an ambulance, they have to wait sometimes as long as 2 hours for an ambulance to arrive because the first responders are so inundated. And Missouri, I have a friend that's a respiratory therapist in Springfield, Missouri, and that they're totally out of ventilators and things like that. They've had to rent them. So again, fortunately, up where I live, up in Pennsylvania here, we seem to be doing fairly well because we have a higher vaccination rate than some of these states. But this is a week old, and I can tell you, if I had a current picture, there's red areas now in the Philadelphia area, New Jersey, and down in Delaware. So it's spreading throughout the whole country. It's more contagious than other virus strains. That one person infects 5 to 6 individuals. The original virus was 1.5. Okay. Unvaccinated people are very high risk for illness. 99.5% of the ICU admissions are unvaccinated. It could be hyperlocal outbreaks, and we see that. And this is what I'm saying. You could argue the point in the United States right now. We don't have a COVID pandemic currently. We have a COVID endemic, meaning that there are areas that are extremely surged with COVID, and there's areas that have sprinkling of COVID. Like in our ICUs, we have about 7 patients that are COVID positive, 2 on ventilators and some on high flow oxygen. I go to Missouri, they have 60 some ventilated patients, and they don't have, their ICUs are all COVID patients with Delta. There are still more to learn about Delta, because Delta doesn't seem to be exactly the same type of virus load and the virus characteristic in the sense of pathophysiology that the earlier coronavirus was. And again, there's no treatment. And it appears that if you get vaccinated, it still is the best protection against Delta. In the Israel data, it showed that with a booster vaccination, that the vaccine, the RNA vaccines like Pfizer, Moderna, were extremely effective against Delta. And there is some talk in the country about getting boosters. And I actually got a booster on Thursday because I was on, last Thursday, because I was in the Pfizer trial. So they asked me if I would be in the booster trial. And I said, sure. I'm still waiting to grow hair in my head, but that hasn't happened yet. Fauci doesn't think that we're going to have lockdowns because, again, there's going to be areas, as the area I live in, that this is going to be a sporadic outbreak. But if you're talking about, places like Florida and that, where where did you see they had 21,000 cases in one day, you may have to have lockdown in order to control it. Now, there's also concern about this being in animals and dogs, especially. They're talking about this canine coronavirus that was detected in Malaysia, and it's coming from dogs. So there's a concern that there's another coronavirus out there. I know that some of the zoos throughout the country are vaccinating animals that could be susceptible to coronavirus. So we could have, in some areas, animals vaccinated, but human beings not. So canine pneumonia is that, there's a concern that this could be a dog virus. That they found that 8 out of the 301 patients that developed this mysterious respiratory disease had the same upper respiratory tracts, were infected with the same viruses that are seen in dogs. Okay, so I haven't heard much about this, but I thought I should add this into the presentation to make people not be able to sleep another night. There's also talk that that you can get infected apparently by multiple coronaviruses. This was a lady, a 90-year-old lady, that was infected with both the UK and the Southern African COVID-19. Her medical history was unremarkable, and she was actually admitted to the hospital after some falls. She tested positive for COVID-19, and she lived alone and received some nursing care at home. The patient had good oxygen, however, she developed worsened respiratory symptoms, and within 5 days she was dead. And when they looked at her respiratory samples, she had evidence that she had both mutants viruses. So now it appears that, you you could be affected by multiple different coronaviruses. Now, when you compare this pandemic to the Spanish flu pandemic, you can see that there was evidence of some kind of mutation. Because if you look at this dotted line, this is your typical type of flu death pattern. Means that either the very young die or the elderly. Okay, most viruses have this this U-shaped pattern where the young, healthy individuals, the death rate is pretty minimal. But if you notice here, there was a spike in 1918. So again, this was first recognized in 1911. Okay, the virus looked like it was fairly stable, and there was some evidence of mutation in 1918. But you can see the amount of younger people that died increased dramatically. Now, some argue that could be also because of World War I was being fought, and living conditions were very, very dismal in Europe and other parts of the world. And there was a lot of malnutrition. But again, this does show that there could have been some mutation that occurred, which made it much more lethal in 1918. And again, you look at these pandemics over the years, and you can see there was 50 million deaths with the Spanish flu. That's about almost over 2.5% of the world's population at the time. All right. So you think things were bad now. We're well over 1 million deaths at this point. This was August 18th. This was as of August 18, 2020. But again, that's not compared to 50 million deaths. And then you look at other things that you thought was bad, like H1N1, which, it seems like I've lived for a lot of these. The only one I wasn't around for was a Spanish flu. Everything else I was around with, so I guess unfortunately I'm still alive. But again, the Hong Kong flu, which I remember this, a lot of people were very sick with this. 1 million people died. All right. So this pandemic, although it's not as bad as the Spanish flu, it definitely is much more lethal than the precursors over the last few decades. And some of these still continue to kill. Okay, like Hong Kong flu still. And that's why when you get the flu vaccine, you get H3N2. Okay. So again, when you look at the difference between the two, actually, the coronavirus has a much higher transmission level than the Spanish flu did. But again, the symptoms are somewhat equal. But it's always, that's your comparison, because that's the last really recorded pandemic at this point. Now, I just want to end with just this type of thing here about where did this come from? All right. So we do know that coronaviruses has been living in bats for decades. And one would argue the point that we've had coronaviruses and common colds and stuff like that. So there are 68 coronaviruses with the branch of the virus tree leading to SARS, COVID-2, that were identified in bats between 1948 and 1982. All right. So there is a great potential that the coronavirus somehow originated with bats. Now, I'm not going to blame the bat and saying that the bat is responsible for COVID-19. That would be totally incorrect at this point. And bats have a very, very important ecological function. There are a host of coronaviruses that have been seen in humans, okay? And there has been other coronaviruses that have caused somewhat epidemics, but never manifested into pandemics. One being the MERS and one being the SARS, which both were very lethal and were very aggressive, but died off extremely quick, which argues the point that did they evolve too quickly and became too lethal to kill off their host too quickly? MERS had almost a 50% death rate. How do you get it from animals to human beings? Again, we talked about this in an earlier slide. So you got these bats that come in, these palm trees, okay? And then there's baboons that climb the trees. And then there's all kind of wind and stir up. And then you have these bats roosting by where people live. And, you know, it's not uncommon for this type of, you know, transmission, in the sense that the bat infects a baboon, a camel, or a cat who comes in contact with humans. Humans themselves can inhale this. They can self-inoculate by rubbing their eyes, picking their nose. They can ingest it in food. And then, you know, again, you can see how you can go from zoonotic to animal host to human being. It's a whole plethora. Like camels, people, you know, get milk from camels. They live with camels. They sleep with camels, things like that. And over the years, again, you look at the MERS seemed to go from bat to camel. The SARS went from this civet cat to a swine to a human, and I believe was a bat, okay, to an immediate host, which now I don't, this was a SARS, which was a precursor for respiratory syndrome. So this was the first COVID we had. As I said, it had the potential to cause a wide world pandemic, but again, it didn't really manifest. It stayed, majority of the transmission and cases were in Asia. They think it came from a cat. Let me just go to your slide here. So here you looked at the number of SARS and the number of deaths, and you can see that this is primarily an Asian disease, but there was some transmission to the United States and to Canada. And very interestingly, they can actually trace where and how this happened. What happened was that the individual that had, you would call this patient zero, they went from their room, went through his hall, and down the elevator. And what happened is that there's, where this red is, there was evidence of SARS environmental you know, where the individual maybe coughed, they touched a side rail, they pressed an elevator button, something like that. So the question would be at this point, how was this, how did this get transmitted? And then how did this become a global pandemic? Well, see, the blue rooms were all individuals that ended up getting infected with COVID, with SARS, and the white rooms were individuals that did not. And what happened was this individual became sick, but these individuals, before they showed their symptoms, ended up leaving this hotel in Singapore and traveling throughout the rest of the world. And you can see that one individual went to Toronto, okay, a flight attendant went to Singapore, another one traveled to Vietnam, and etc. So you can see how quickly you can spread a virus from a very remote, distant area to other areas throughout the world. And that's how SARS were spread. And SARS is the same kind of symptoms as COVID. I don't need to, really kill this. They call it respiratory problems. A death, this is a patient that died of SARS, and they have their hemorrhagic lung. Too virulent, killed off its host too quick, mutated to extinction. Again, was it a practice run for the COVID virus, saying, let's try this and see what happens? This failed, so it came back to fight another day. Then there was MERS, which was recalled Camel Flu, which had very much interest because it occurred the same time we were over at, we had the Iraq war was going on, so we had soldiers that were, a very strong military present over there. And I believe it went from transmitted from bat to camel to human. And the symptoms were the same as, COVID. It's, and there was no vaccine, no medicine for it. And I don't want to go through because I'm running out of time here. But again, in the United States, there was only 2 cases. Both of those individuals had been working in Saudi Arabia, and they believe they must have transmitted it from, traveled from Saudi Arabia to the United States. Both of those individuals, however, were hospitalized, but they ended up doing okay, and there was no deaths. And again, MERS kind of died out as quick as it came, but it was very lethal. Now, the evolution is very interesting because theory 1, and there's 2 theories. Theory 1, it came from bats, and these bats carried this COVID and it could have infected the cat, the Pelargonian, which is kind of this like anteater that has an armored body. So the horseshoe bat is probably the one that takes the most responsibility for spreading COVID, because it is very common in China, and a lot of the caves where COVID has been kind of identified, COVID-19, was in the horseshoe bat caves. And again, they don't really know totally which animal is responsible for going, again, bat didn't infect human, infected one of these different type of animals. So so again, this was MERS, this was probably SARS, and they think the Patagonia was responsible for COVID too. But this bat doesn't infect a human, infects one of these animals who then transmits it to human, because the human doesn't hang around with bats unless you're in a bat cave, as Bruce Wayne. But the rest of these animals do have close association with human beings. And there is a Patagonia, and these poor things have been slaughtered unmercifully because they've been believed to be the carrier of COVID, which is truly despicable. And the belief is that the Chinese wet market, where a lot of these animals were sold and there's tremendous amount of people, was where this actually spread very rapidly from infected animal to human beings. And again, there's a lot of these animals there. The human beings have lots of contact with these animals, and they even sleep with them. So the Chinese wet market really took the brunt of being the major transmitter of COVID-19. And again, it's been that Patagonians are sold for food and live animal wet markets, and they believe that's the way we spread, okay, is from these bats to this Patagonian to individuals, and lots of individuals who had interactions at these wet markets, okay. Now, so that's theory 1. Theory 2 is that it was an escape virus, the Wuhan theory. And I think Wuhan Institute of Virology was near the epicenter and was located about 2 blocks from a large group of wet markets, okay? And so what's kind of interesting is that there was all of a sudden, out of nowhere, in the end of 2019, these bad, mysterious pneumonias, and they were spreading through the lab, spreading through Wuhan, And one of the virologists at Wuhan lab, Xing Ling, okay, said that she was very concerned because she was afraid that this virus may have come from her lab, okay? And there reports that researchers at the institute were performing some gain of function experiments, which you actually use, you take a natural virus like the corona, like the SARS virus, and you modify it to become more virulent or better to infect humans. Now, I'm not saying this was being done as a way of germ warfare, but the bottom line, this is done in the United States too, so it's not done, it's just done in Wuhan, is the bottom line is that sometimes you do this to try to figure out how you can find a weakness of the virus if it mutates to a stronger level. So the problem here is that, one, this Wuhan seafood wholesale market was the blunt of the responsibility, saying it came from there. But to this date, there's no infected animals were identified, and Chinese researchers have ruled out that the origin of virus did not come from these animals, but they think it may have come from the lab at this point. And interestingly, in December of 2019, this is what the Wuhan Institution of Virology looked like. You couldn't get within, near it because it was, that's a pretty armed, that's a pretty much of a fortress. You don't have more than, 6 feet between the soldiers there. So no one's going to get in there at all at this point. So there was a true event that occurred at the Wuhan lab that needs to be really, really kind of, drilled down a little bit more. Now, this is what really where I think the bats and the Wuhan escape comes from, is that I think that there were 6 miners who were hired to clear the cave of bat guano. Bat guano is bat feces that is extremely rich in nutrients and is used for fertilization. And there has been a huge, huge push for China to improve their production of produce and gardens and things like that. And what happened is 6 of these miners developed severe respiratory decompensation that was very similar with SARS. Several bats that were captured were infected with coronaviruses, and 3 of these miners died of COVID-19 like pathophysiology. So what happened was the China virology from Wuhan came to this lab. They shoveled up specimens and went back to the lab, and it looks like that somewhere this original virus escaped from the lab that then caused caused leakage. And everybody in the lab has safety training and psychological evaluation and stuff like that. So the argument from China has been that they didn't keep these live viruses, but the people that worked there who kind of escaped that lab before it was locked down said they did have these viruses. So I truly think that these viruses were taken from this cave to Wuhan Institution of Virology to be studied or whatever, and somewhere they escaped. They got into the wet markets where there's tremendous amount of people, and that's where this origin. That's kind of what I think based on what I have read. This is what the individuals now look like working in there, and that's pretty protective, I would argue, the point. So one would argue that, there has been US science call for fuller probe. Joe Biden has said that, he's formed a committee to evaluate this because so the theories of accidental release from a lab and zoonotic spillover both remain viable. Nobody has argued that one theory is more acceptable than the other, but I have to believe that something happened in that lab because that lab has been off limits to really anybody from the outside since this pandemic has occurred. So we really don't know. And no matter who you talk to and what side you take, it won't be satisfied. So I'm going to stop here with this presentation, and I'd like to go on to the pathophysiology presentation.